Today, we're learning about mitosis and meiosis, or more simply, cell division. I'm sure you've heard of these before, but I'm here to break it down. Be grateful that you have mitosis, or you wouldn't be able to grow, replenish your cells, or even survive! There are 3 things you need to know about mitosis. 1. There are 6 steps: interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. 2. Every daughter cell that is split is identical to each other. 3. Mitosis is not the same as meiosis. Let's go over the steps of mitosis. Let's go over the steps of mitosis. Interphase. Interphase is pretty much the preparation for the cell to divide. The DNA is unraveled and duplicated. Prophase. During prophase, the DNA is condensed and centrioles appear. In between prophase and metaphase is prometaphase. This is when the nuclear envelope dissolves to allow the chromosomes to be latched onto and pulled apart by the microtubules organized by the centrioles on the sides. The microtubules attach to the cell's kinetochore. A kinetochore is a type of protein complex that the spindle microtubules are attached to, assembled on the centromere. A centromere is a region of highly condensed DNA in the approximate middle of each chromosome. The centromere holds 2 sister chromatids together. Sister chromatids are exact copies of each other, which happens when the DNA is replicated. Metaphase! Now that all the microtubules are attached, the chromosomes are lined up in the middle of the cell, or the metaphase plate, so they can be pulled to the opposite poles of the cell. On either side of the middle are identical sister chromatids. This is so the resulting cells have the exact same DNA. Anaphase. The centromeres are dissolved, and now the sister chromatids can be separated. Telophase. Here the chromosomes can finally move to their respective sides via microtubules, and a cleavage furrow is formed. Cytokinesis. In cytokinesis, the cleavage furrow is the cytokinetic furrow, and an actin-myosin contractile ring is established right in the middle. This contractile ring contracts, and one cell is now 2. Well, we near the end of our cell dividing journey, I kind of miss it. Not so fast! I said, not so fast! We are not done yet. Remember number 3 of the 3 things that I told you needed to know? Mitosis is not the same as meiosis. If you forgot that, I don't really know what to tell you, but it's like a minute ago, but mitosis is not the same as meiosis. Another process of cell division that occurs in sex cells. But what about meiosis makes it different? There are 3 things you need to know about meiosis. Can you tell there's pattern and structure to the video here? Is it just repetitive? Because I always welcome constructive criticism. 1. 2 cycles of division. 2. Crossover. And no, I'm not talking about your 2 special interests having a semi-canon interaction on Forge. And 3. Independent assortment. But wait, what was that? Did you say 2 whole cycles of division? First, meiosis 1, pretty standard. Interphase, prophase 1, prometaphase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis. But get this, meiosis 2!
Huh?
Interkinesis, prophase 2, prometaphase 2, metaphase 2, interkinesis, prophase 2, prometaphase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. Again. That's so much. Oh my god, if that was me, I'd die. Well, actually, that was me. And it's actually the reason I'm alive, so I guess not. We can just move on. Let's just move on. Well, it starts the same as mitosis with interphase, which by the way has 3 parts. I probably should have mentioned that before, but it didn't really matter then. We're going hardcore now with meiosis. Interphase has G1, S, and G2 phases, but all you really need to know is that DNA duplication happens during the S phase. Now, prophase 1 is where crossover starts. Homologous chromosomes are paired together and tightened by the synaptonemal complex. Yeah, it's just a layer of proteins between them. They're so tightly paired that even the individual genes are aligned together. Wow. Now, keep an eye out, because something cool is about to happen here. Let's do a little zoom in. What's that? They're sharing genetic information? Well, more like swapping individual genes with each other. As prophase goes on, the synaptonemal complex, dissolves and the homologous chromosomes move apart again. Prometaphase is pretty much the same, but metaphase 1. Oh, that'll take you for a doozy. Metaphase 1 is where independent assortment happens. It's pretty much true to its name. It just means the chromosomes, while they're lining up, are put in a random order. You know what this means? Huh? Once the cell divides, each cell's genetic information is unique. Woo! That, along with crossover, is how we get genetic diversity. Wow. That's how this guy turned into that guy. Hello. And this guy turned into this guy. Anaphase 1, it's just where the microtubules pull the linked chromosomes apart, yada yada. Telophase 1, chromosomes at opposite poles, cytokinesis does its thing, but what's up with the chromosome count? Haploid, diploid. Let's take that to our thinking corner.
Welcome to the thinking corner. Today we're counting how many chromosomes are in the cell during mitosis, meiosis 1 and meiosis 2. Let's start with mitosis. Mitosis starts with a diploid cell, meaning it has 2 sets of chromosomes. Mitosis ends with 2 diploid cells, each with 2 sets of chromosomes. Meiosis starts with a diploid cell and ends with 4 haploid cells, meaning each cell has 1 set of chromosomes. 1 set of chromosomes is 23 chromosomes. 2 sets of chromosomes is 46 chromosomes. Sex cells are haploid, and the rest of the body's cells are diploid. Back to you.
Thanks, Queen Lizzie, for making time for us in the afterlife. May she rest in peace. Anyways, on to meiosis 2. What chromosome count are we at? Do we know? Does anybody know? Okay, so we have 2 haploid cells, and we need 2 more. So let's do it again. Interkinesis. That's new. Interkinesis starts off meiosis 2 with a quick interphase, but you know how we were talking about the G1, S, and G2 phases? Well, interkinesis doesn't have an S phase, so there's no more duplicating DNA. No. So as we go on to prophase 2, each cell has 1 set of chromosomes, but each with their own sister chromatids. So the chromosomes really look like an X right now. Prophase 2 just gets the cell ready to divide again, dissolve the nuclear envelope, form in the spindles, just putting everything in place. Prometaphase 2, no more nuclear envelopes, spindles are ready, kinetochores and all that jazz. And then metaphase 2, they all line up again, and now we're at anaphase. In anaphase 2, the sister chromatids are pulled from each other. You hate to see a family torn apart, but don't blame me, blame the microtubules. Telophase 2, chromosomes are at opposite parts of the cell, dramatically screaming for their sister back, but oops, too late. Cytokinesis is here, and you're never seeing them again. So now we've got 4 cells, each with 1 set of chromosomes, and all of them unique. And that, folks, is meiosis and mitosis! Thank you! Oh, this was way too long!